This protocol is significant because it provides detailed instructions on how to prepare and inject mosquito embryos for CRISPR-Cas9 genome editing.
The main advantages of this microinjection technique are that it has a high rate of mosquito survival and allows researchers to determine the function of a gene.
This protocol has been specifically tailored to inject mosquitoes that belong to the genus of Culex. These mosquitoes transmit several pathogens, including the viruses that cause West Nile fever and St. Louis encephalitis, as well as filarial nematodes that cause canine heartworm and elephantiasis. Therefore, having tools such as CRISPR-Cas9 genome editing will allow us to better understand and control these important disease vectors.
Injecting freshly laid insect embryos and getting a high rate of survival takes lots of time, practice, and patience. Therefore, we recommend injecting several embryos with a colored saline solution before injecting expensive materials to generate a mutation.
Begin by backfilling the injection needles with the injection mix. Choose a needle filler or gel loading tip that will fit easily into the back end of the selected injection needle. Make sure that there is a bit of space between the end of the needle filler and the injection needle. Carefully aspirate a single, small drop of the injection mix into the injection needle, near the spot where it begins to taper. Then, attach the injection needle to the microinjector. Place the slide containing the embryos onto the stage of the microscope. To open the needle, use a starting injection pressure of approximately 30 psi and adjust it as necessary to deliver an